Welcome to the PubMed Daily Review for March 08. Today we scanned 2,482 new open access papers. Here are the top 5 selected for detailed analysis. Up next, our first paper out of 5, PROTAC-Mediated Degradation of mHTT Aggregates Attenuates Neurotoxicity in Cellular and our 6/2 Mouse Models of Huntington's Disease by Pachao Liu and Joseph Jinsei Huang from Institute of Chemistry, 38017 Academia Sinica, Taipei, 115, Taiwan. Let's break down its key contributions. Huntington's disease is a devastating inherited neurodegenerative disorder. It's caused by a genetic mutation that leads to the buildup of toxic protein clumps in the brain. Current treatments struggle to effectively target these toxic proteins without harming healthy cells. This research investigates a new technology called PROTACs, designed to specifically break down these harmful aggregates. To tackle this challenge, the researchers developed a specific PROTAC molecule, called PROTAC-2-foot. They tested its ability to degrade the toxic mHTT protein in both lab-grown cells and a mouse model engineered to mimic Huntington's disease. This PROTAC works by tagging the harmful mHTT aggregates for destruction by the cell's own machinery. They then assessed if this degradation reduced toxicity and improved disease symptoms. The results were very promising. PROTAC-2-foot effectively and selectively degraded the toxic mHTT aggregates without affecting healthy huntingtin. This led to a significant reduction in cell death in their lab models. Importantly, the PROTAC was able to cross the blood-brain barrier in mice. In the R6/2 HD mouse model, treatment with PROTAC-2-foot improved motor function, increased survival time, and reduced key markers of disease pathology in the brain. This study demonstrates that selectively degrading toxic protein aggregates is a powerful therapeutic approach for Huntington's disease. PROTAC technology offers a novel and potentially more targeted way to treat HD compared to existing methods. These findings open exciting avenues for developing PROTAC-based therapies not only for HD but also for a range of other neurodegenerative diseases caused by protein misfolding and aggregation. Up next, our second paper out of 5, Comparative Efficacy and Safety of Glucagon Receptor Agonists on Metabolic Outcomes. A network meta-analysis of randomized controlled trials by Aya Abulehia and Barbara Gisela Carranza Leon from Faculty of Medicine, Al-Quds University, Jerusalem, Palestine. Let's break down its key contributions. Obesity and type 2 diabetes are closely linked and represent a massive global health burden. Achieving significant weight loss is key to reducing the risk of serious complications like heart disease. While drugs like semaglutide have shown promise, new classes of medications, called glucagon receptor agonists, are emerging. These drugs work differently, potentially offering new avenues for treatment. To compare these new drugs, researchers conducted a network meta-analysis. They systematically searched major databases for randomized controlled trials involving 4 specific glucagon receptor agonists: retatrutide, survodutide, mazdutide, and cotaglutide. The analysis included data from 14 trials with over 3,000 participants who had type 2 diabetes, were overweight, or obese. They compared these drugs against placebo, focusing on weight loss, HbA1c reduction, and safety. The results were quite striking. Retatrutide led to the most significant weight loss, averaging over 13 kilograms compared to placebo. It also showed the greatest reduction in HbA1c levels. However, retatrutide and cotaglutide were associated with higher rates of discontinuation due to adverse events. Mazdutide, on the other hand, demonstrated the most favorable tolerability profile, with the lowest discontinuation rate. So, what does this mean for patients? Retatrutide and survodutide appear to be highly effective for managing weight and blood sugar in individuals with type 2 diabetes and obesity. However, it's crucial to consider the side effect profiles, as some drugs are better tolerated than others. While this meta-analysis provides valuable comparative insights, direct head-to-head studies are still needed to definitively establish the best treatment choice for individual patients. Up next, our third paper out of 5. Inhibition of NAMPT targets DNA damage response to sensitize alkylating chemotherapy in TP53 mutant mantle cell lymphoma by Na Li and Hong Yang from School of Life Sciences, Liaoning Provincial Key Laboratory of Biotechnology and Drug Discovery, Liaoning Normal University, Dalian, China. Let's break down its key contributions. Mantle cell lymphoma, especially with TP53 mutations, is a tough cancer to treat. Patients often don't respond well to chemotherapy and their disease progresses quickly. We need new strategies. NAMPT, an enzyme involved in NAD+ metabolism, is found at high levels in MCL and could be a promising target. To investigate this, researchers used mantle cell lymphoma cell lines, both with and without TP53 mutations, as well as mouse models. They tested a NAMPT inhibitor called KPT-9274, both by itself and in combination with a common chemotherapy drug, bendamustine. The goal was to see if this combination could overcome resistance and understand how it worked at a molecular level. The results were very encouraging. The combination of the NAMPT inhibitor and bendamustine showed significant synergy, meaning they worked much better together than alone. This led to profound tumor regression in the mouse models of TP53 mutant MCL. Importantly, this enhanced effect did not come with increased toxicity, suggesting a favorable safety profile for this combination. This study highlights NAMPT inhibition as a potential precision medicine approach for TP53 mutant mantle cell lymphoma. By targeting NAMPT, we can sensitize these resistant cells to existing chemotherapy. The findings strongly support moving forward with clinical trials to test these combinations in patients, guided by their TP53 mutation status, to address a significant unmet need in this challenging lymphoma. Up next, our fourth paper out of 5, beta-integrin expression defines functionally distinct CD4+ T-cell subsets that are altered in ulcerative colitis by Kirsten A. Ward-Hartstang and Pedram Tevakoli from Department of Surgery, University of British Columbia, Vancouver, British Columbia, Canada. Let's break down its key contributions. Ulcerative colitis affects millions worldwide, and T-cells play a key role in its gut inflammation. While treatments like vedolizumab target alpha-4 beta-7, many patients don't respond well. Interestingly, higher levels of alpha-4 beta-1 plus T-cells are seen in non-responders. This raises an important question: do these different integrin-expressing T-cells actually behave differently in UC? To investigate this, researchers analyzed T-cells from gut biopsies of UC patients and healthy controls. They used advanced flow cytometry to look at specific T-cell markers, focusing on the expression of alpha-4 beta-1 and alpha-4 beta-7 integrins, and measured the production of key inflammatory cytokines like IFN-gamma and IL-17. This allowed them to compare the functional profiles of these T-cell subsets in the context of UC. The study found significant differences in T-cell populations. UC patients had increased regulatory T-cells and, importantly, a lower frequency of IFN-gamma producing T-cells. They also observed a higher ratio of beta-1 to beta-7 integrin expression in UC gut T-cells. This ratio was strongly linked to cytokine production, showing a negative correlation with IFN-gamma and a positive one with IL-17A/F. This research reveals that beta-integrin expression isn't just about T-cell homing, it's intrinsically linked to their function and cytokine output. The shift towards beta-1 expressing cells, which are enriched for IL-17A/F, appears to be a key feature in UC. These findings open exciting possibilities for developing more targeted therapies that specifically modulate these distinct T-cell subsets. Up next, our fifth paper out of 5. Lipid donor-anchored genome mining uncovers dioxinopeptins, antibacterial lipopeptides with a 1,3-dioxane functionalized polyunsaturated lipid tail by Ying Chen and Yiming Shi from A. School of Pharmaceutical Sciences, Shenzhen, Sun Yat-sen University, Shenzhen, China. Let's break down its key contributions. Lipopeptide antibiotics are powerful medicines, but finding new ones has been challenging. Most research focuses on the peptide part, often overlooking the lipid tail, which significantly impacts how well the antibiotic works. This study introduces a novel strategy to discover these important compounds by looking for the genetic machinery that builds the lipid tails first. The researchers developed a new method called lipid donor-anchored genome mining. They used genes for polyunsaturated fatty acid synthases, PUFAs, as a starting point to find gene clusters that also produce non-ribosomal peptide synthetases, NRPS. This led them to identify a new family of gene clusters, the DXP family. They then cultivated bacteria containing these clusters, isolated the compounds, and used advanced techniques like mass spectrometry and NMR to determine their structures and functions. The study successfully identified dioxinopeptins, a novel class of lipopeptides. These compounds showed selective activity against methicillin-resistant Staphylococcus aureus, or MRSA. Their mechanism involves disrupting the bacteria's ability to produce energy by interfering with proton motive force and ATP production. A key feature is their unique lipid tail, functionalized with a 1,3-dioxane ring, which is added through a post-assembly modification step involving the DXPH enzyme. This research demonstrates that focusing on lipid donor genes is a powerful strategy for discovering new lipopeptide antibiotics. The unique 1,3-dioxane functionalization on the lipid tail of dioxinopeptins is critical for their activity against MRSA. This work significantly broadens our understanding of lipopeptide diversity and provides a valuable new avenue for developing much-needed antibacterial agents.